So I want to talk to you today about genomic analysis of speciation and adaptation in Aquilegia. We've been, like Dan said, we've been working on Aquilegia. I've been working on almost 20 years now, since my postdoc days. And we were really fortunate to convince and collaborate with JGI to get a genome sequence. So I'll be talking a little bit about that today as well. One of the big things about Aquilegia here that I should point out first off is that the unique feature of the genus is that each petal has a long tubular outgrowth, and it's just a long, slender, hollow tube with a nectary at the tip that produces nectar that fills up the spur, and then pollinators have to probe down the spur to reach the nectar, and in doing so pollinate the plants. Another reason, or one of the big reasons that we put forward for why we should sequence Aquilegia is that it has a really unique phylogenetically important position in the angiosperms. And here on phytosome, we see the species that are sequenced on version 6 here. And then here's another phylogeny for the angiosperms. Most of the species that have been sequenced are down here in the monocots. So like this branch down here, this is the branch for the angiosperms on phytosome. And then things up here in the core eudicots. But really nothing in between here. And these are very deep phylogenetic branches in the angiosperms. And these are these weird, unusual basal lower eudicots here. And Aquilegia is among the group that is at the very base of that. So this is a key position, almost equidistant phylogenetically between the core eudicots and the monocots. So we're hoping that it'll spur some genomic analysis for genome evolution within the angiosperms here. I won't talk so much about that. On the initial sequence, we have about a 300 megabase and just under 1,000 scaffolds. I'll talk a little bit more about that later. So the genus is about 70 species. It's found throughout the northern hemisphere. There are European, Asian, and North American taxa. On this slide, we have the top row and the bottom row are some of the taxa from North America. This is an Asian species, fragrans, from Kashmir, and this is a European species over here. So they have this incredible floral diversity, and as I'll show you, that's correlated with pollinator types and adaptations to different pollinators. The other striking thing about the genus is that virtually any pair of these species can be crossed and fertile hybrids can be made. So we can do genetic dissection of any of these traits that differentiate these species. This is really a remarkable feature. We can even cross European and North American taxa to get fertile hybrids. They also differ not only in this floral morphology, but in their ecological adaptations. We have a lot of the species are in montane habitats, but they also occur in desert regions. Here, I'll talk about this habitat here. This is the Serpentine seep area just on the other side of the bay here. And this is up in Wyoming. I'll show you some of these later. All of these pictures actually have columbines in them, but they're a little bit hidden away. So we'll come back to those adaptations to habitats and soils in a minute. So again, a couple of things about Aquilegia. I want to get to the adaptation and speciation, but there are some broad macroevolutionary patterns that are really interesting in Aquilegia. And the first is that the evolution of these nectar spurs we found across multiple independent origins of nectar spurs in the angiosperms. So nectar spurs have evolved many times in the angiosperms. And almost always they're correlated with an increase in species diversity. So that when spurs evolve, we get a burst of speciation, or at least diversification in that lineage. And that's one of the best characters for any organism for the evolution of a particular trait and an increase in species diversity. There's also directional trends that we've discovered in the evolution of some of the particular traits. One is that spurs tend to get longer during evolution, but they do so by shifting to pollinators with increasingly long tongues. And so what we find in the phylogeny is that bee-pollinated, short-tongued bee-pollinated species are at the base of the phylogeny. Hummingbird pollination evolves from bee pollination, and then hawk moth pollination evolves from hummingbird pollination. And that trend, we don't get reversals and we don't get skipping across this way either. And most of this spur evolution is concentrated at the time of speciation. So you can imagine that if you shift to a different pollinator, even though they're intercompatible, because the pollinators only visit one species or the other, they're enforcing reproductive isolation. And so this switching of pollinators, changing in spur length, is concentrated at speciation events. Okay. Again, these transitions have occurred multiple times. So this is a phylogeny for the North American columbine. And here we've color-coded the tips here. The blue are being bee-pollinated species, the red are hummingbird, and the yellow are hawk moth pollinated species. We have transitions from bee to hummingbird here, and then multiple transitions from hummingbird to hawk moth at each of these arrows. Another point to make out in this phylogeny is that this is based on AFLP markers, about 1,600 AFLP markers. The species are incredibly closely related. Most of these polymorphic AFLP markers are polymorphic across species boundaries. So only 1% of the markers are clade defining within one of these clades here and signify a clade. So most of the variation is shared across taxa, and I'll come back to that in a bigger way in a little bit. Okay, so now we can focus in on one species pair to ask what are the traits that are important for reproductive isolation, and then I'm going to show you how they're important in reproductive isolation and then move towards identifying the genetic basis for those traits, which is one of our big goals in our lab, is to identify the genetic basis of these adaptations and traits that are important in speciation. These two species are historically really important. Vern Grant used them as his first example of how pollinators could provide reproductive isolation in plants about 60 or 70 years ago now. Aquilegia formosa grows along the west coast of North America, here in California, all the way up to Alaska. And this species pubescens, though, only occurs in the high elevation of the southern Sierra Nevada, from about Yosemite down to Kings Canyon National Park. The two species obviously are really different in flower morphology, and they're different in their habitats. Formosa occurs along stream sides, in shady, forested areas, in really well-developed soils, while pubescens is above tree line in the alpine in these really poor soils, developing in these really rocky granite outcrops here. The species are visited by different pollinators. Formosa is visited a lot by bees and a lot by hummingbirds. We don't think bees are very good pollinators because they're, as we saw earlier today, they're actually foraging for pollen, and the flowers go through a male phase and then a female phase. The bees avoid the female phase flowers, so they're not very good pollinators for them. While hummingbirds and hawk moths are foraging for nectar, and they visit both male and female phase flowers. So, and then pubescens has a lot of hawk moth visitation, but virtually no hummingbirds. And again, the formosa, the red one, has lots of hummingbirds, but essentially no hawk moth visitation. Now, where the two habitats come close together, natural hybrid zones can form. So right about at tree line in the Sierra Nevada, you can find areas where you have rock slides, which is pubescens habitat, and stream sides where there's formosa habitat. And in the areas in between here is where we find natural hybrid zones between the two species populations. And we find this incredible diversity of floral morphology in those zones. So this is all individual pictures from a single zone that I just showed you. And here's a hawkmoth visiting a hybrid in that zone. So I had a student ask, well, when the pollinators are given the two species, they're really good at deciding which species to go to. Hummingbirds really like the red one, hawkmoths really like the white ones. But the question is that when reproductive isolation is breaking down and we get this diversity here, is there still assortative mating in those populations? And so what he did was to go into this hybrid zone and map every single plant in the population. He measured flower morphology, which way the flowers point, the colors of the flowers, and the lengths of the floral parts. And then he did a paternity analysis. He collected seeds off of every plant, genotyped the seeds and mom, figured out who dad was, and since he'd measured all the plants in the population, he knew what dad looked like. And he could ask whether plants that look similar to one another are more likely to mate with one another, even in this sea of diversity, where was reproductive isolation and assortative mating still occurring. So here's a map of the population. Blue are plants that look pretty much like pubescens, red for formosa, and yellows for hybrids. And you can see hybrids are scattered out, particularly through these three areas here. He used a logistic regression to ask whether any pair of plants, what their probability of mating was. And the idea here is that as plants are close together, they should be more likely to mate. So as distance increases, the probability of mating should go down. And if assortative mating is happening, the same thing. As the plants are increasingly different in their flower morphology, the probability of mating should go down. And this is exactly what he found. So the floral characters that were important are the length of the spurs, which way the flowers point, and the color of those flowers. So the more similar plants are in their spur length, in their orientation of their flower, and their color, the more likely they are to mate with one another. And so he could build a mating surface for this population. And the formosa, this is a measure of the flower morphology of moms and the flower morphology of dads. This is where a high probability of mating is when there's two formosas or two pubescens, and the interspecies are really unlikely to mate. So even in this great diversity of flower morphology, we're still seeing reproductive isolation, or at some degree, because of assortative mating. And so the question for us now is, what's the genetic basis of these traits? Okay. So we've done QTL mapping on the genome. As I said earlier, the genome's only about 300 megabases. It's a nice small genome. There's only 7 chromosome pairs. And we've made a initial map here mostly with SNPs to ESTs and some microsatellite markers here. And when we do a QTL analysis, we can find QTL a fairly large effect for the traits that we're interested in: flower orientation and spur length. And flower color, as it turns out, there's a single very large QTL on what we call linkage group 7 here. Now, we've focused in on flower color for a couple of reasons. One is that it's one great big QTL, which makes things easier. And then, of course, we know a lot about the biochemistry of flower color, so we have a lot of candidate genes to focus in on as well. So, here's the nucleus and the cytosol and the vacuole, how flower colors are made. Most of the reds and blues of flowers are anthocyanin pigments. And there are these 6 main enzymes taking the biochemical precursors and moving them down the pathway this way, where they're modified slightly and then pumped into the vacuole, where they accumulate and give the flower color. And then from other systems, we know a lot about the transcription factors that control the expression of many, if not all, of the genes in this pathway. So when we look at these two species, what we find is these first genes, if we just do RT-PCR, they don't differ in these early genes in the pathway, but all of these late genes in the pathway, the white species, pubescens, is downregulated relative to the species that makes the anthocyan and pigment. So as I said, in the F2 population, we have, this is a measure of flower color and just a frequency distribution. We have almost exactly a 3-to-1 ratio of red flowers to white flowers, signifying a single large QTL. And that single QTL, but turning down 3 genes simultaneously, suggests that this is a transcription factor that's causing the difference. And one of these, perhaps up here. Okay. So now once we get to the genomic analysis or the genome analysis, we know that flower color is up here. If we expand linkage group 7, and all of these markers again are on ESTs, we can place these markers onto the scaffolds, and we find that 2 scaffolds, scaffold 15 and 10, overlap where this QTL is here. And we can go in and look at the genes in here and look for genes that are important in the anthocyan and pathway. And what we find is that there is a MIB gene that's right in the middle of this scaffold here. This is phylogenetically related to other MIB genes and other taxa that control the anthocyan and pathway. So it was a really good candidate. We also found one of the structural genes was in there as well. So both of these are correlating with color in the QTL analysis. As someone earlier talked about, that this is a QTL analysis is fairly broad, and so it's lots of genes are correlated at this point. So we wanted to ask whether we could go back in and use the genomic analysis and ask whether we can look basically for association mapping to identify the genes that are important for color in there. So I'm going to show you a beginning analysis that we did. This was a grad student of Magnus Nordborg's that did this work, Liz Cooper. And she sequenced 9 genes for 96 individuals, found all the SNPs in them, asked whether we could differentiate the 2 species. These are completely different species. And here's a structure analysis, and we cannot separate out the 2 species at all after sequencing 9 genes. This isn't a lack of variation. There's lots of variation, lots of SNPs in there. It's just the 2 species have the same allele frequencies, essentially, between the 2. This is actually really good news for us. There's no, most of all of these genes don't show any structure, and what we're looking for are the markers of structure or the differences in flower color, in this case, between the 2 species. Actually, the FST value between the species is essentially 0. And in this analysis, I know it's only 9 genes, but linkage disequilibrium is only on the order of 1 kb. This suggests to find differences between them, we're going to have to find markers right at the point that's really important between the 2 species. So this is really a prime place to do population resequencing and looking for differences between the taxa. So this is what we did here. Here's California. We're somewhere right in here. And this is expanded here. We have 6 populations of Formosa and 4 populations of pubescens. We took 27 to 64 individuals from each population, pooled them together, and then resequenced them to look for differences between the species. We ended up with about, aligning this to our reference genome. We have about 32 reads per base pair, but quite a few places that have still low coverage in this analysis here. And if we look at this graph here, is all this is showing is that what we're really looking for is an allele frequency difference. So we want the frequency in Formosa and the frequency in pubescens, and that this is a large difference between the two species. So that's going along this axis here. So this is a big difference. These aren't different. This is the number of reads per species. When we don't have very many reads per species, then our estimate of allele frequency is really poor. So this is accuracy going in this direction, and this is how many sites are different. And as you can see from this graph, there aren't very many sites that are different between the species. And in fact, if we use just a cutoff of a site having to have at least 30 reads in both of the species, we found a whopping, well, we found over 3 million SNPs, but only a little over 500 of them differ by 90% in their allele frequency difference between the two species. If you go up to 95, there's 69 SNPs, and there are only 43 fixed differences between the two species genome-wide, which is just remarkable. I had been working for 20 years trying to find a fixed difference between these two species, sequencing gene by gene. It was never going to work until we got this done. Sometimes I'm lucky, but I'm never that lucky. Okay, so the number of genomic regions differentiating the species is incredibly small in this case. So let's go back to that genomic region for flower differences. Here's that the QTL. We have a scaffold here with MIB12 and a scaffold with DFR. And if we zoom in on those, as we got the genome sequence, we had this MIB, and we zoom in and we found out actually there were 5 MIBs all grouped together, which we weren't quite expecting at first. But if we look at the differences at those SNP positions, so this is allele frequency difference here, we're looking about 200 kb here, we get this nice peak coming up to MIB12, which was our candidate gene to begin with. And if we look at the other gene that was associated in the QTL analysis, we don't see a big peak around DFR. So this really suggests that this is probably important. And if we zoom in on it, this fixed difference SNP, this is one of the 43 fixed differences between the species. It causes an amino acid change in this MIB gene. Okay. So that's our real candidate now for what's causing flower color differences between these species. Okay. So going back to our phylogeny, one of the points I wanted to make too early on is that we've had these same transitions multiple times, so we can ask whether the evolution of the same trait has occurred through the same manner multiple times. So here's pubescens. We have MIB12 that seems to be downregulating multiple genes. And now we'll look at another species that is polymorphic within the species, the Colorado state flower Aquilegia Coerulea. So in Aquilegia Coerulea, if you look at postcards from Colorado, you'll see this one up here, but it's polymorphic. There are individuals that can be pure white, and the blues here. This is a mostly white population with a blue individual down here. So my student Nathan Derrig has been doing RNA-seq on this. He took sepals. These are the sepals coming out here from 2 polymorphic populations and made pools of blue and pools of white, and then used RNA-seq to look at the expression differences. So we have log fold change here, and this is the probability. On this side is higher expression in blue, and this is higher expression in white. And that's a little dark up here. But if you focus in on this, the highest, most significant SNP that, or expression difference that has a fairly large difference in expression is this gene, F3'5'H, and this is the key gene for making the blue color. The difference between, say, red and blue colors is completely dependent on this gene being present or not. So it's in high expression in the blue, as you might expect, and low expression in the white individuals. Of course, the nice thing about RNA-seq is that we get sequence information at the same time, and he'd done these sequences in 2 different populations. This is all of the scaffolds in the genome lined up, and the difference in the SNP allele frequency between the blue and the white pools for both populations. And only one place is there a highly differentiated SNP in both populations, and it's in the same gene, F3', 5'H. Okay. And then we've zoomed in on this, and this is mostly a difference in the 3' untranslated region, or in the promoter region, which is probably controlling the expression difference. So if we look in one more difference in flower color, this is the line that we sequenced for the genome. This is another independent loss of anthocyanins. The yellow color is carotenoids. If we cross those, we make an F1. Here's an F2 population. This is blooming right now in the greenhouse. And we're going to use this population to unite the scaffolds and unite the physical map to a genetic map. And then we'll also use it for differentiating the color differences. I want to point out in the last few minutes some of the other things that we can do with Aquilegia. Not only are these flower color differences, but even in these very closely related taxa here, there's really interesting differences. So again, we have Formosa, which I talked about earlier, but we have Aquilegia eximia, which grows almost exclusively on serpentine soil. So here across the bay, there are multiple populations where the 2 species grow very closely together. They do not overlap in flowering time. They can be 100 feet apart, and there's absolutely no overlap in flowering time between them. Working with Justin Whittall and some of his undergraduate students on this project, the soil chemistry between these soils is really extreme. The calcium-magnesium ratio is high in the Formosa and really low in eximia, classic serpentine soils, and nickel, which is really bad for plants, is really high in this eximia population here. If you take seeds from the 2 populations and grow them on the soils, Formosa just dies on eximia soil, and eximia can grow on eximia soil. It actually likes the Formosa soil better, and is growing better there. That's another classic serpentine characteristic, that they're probably worse competitors here, and they're forced to live on this eximia soil. That's the hypothesis. Another species, just to point out from the beginning, here's a species that grows on really high calcium soils. This is up in Wyoming with really high soils here. This is Aquilegia jonesii. It's this little columbine that only grows about this tall and pollinated by bumblebees. I'll just blow through real quickly. There's also interesting floral development differences with Aquilegia. There's a 5th floral whorl that most plants don't have. And so sepals, petals, stamens, then the staminodia and carpels. And the staminodia surrounds the carpels here during development. With expression studies, we've found about 160 unique genes, and these all seem to be important with herbivory defense. And these may be protecting the carpels during development. There's all sorts of other interesting floral differences between them. I'll zoom through those. We have some new techniques, or some techniques developed by Elena Kramer to do basically RNAi with VIG systems, so we can turn off genes and we can turn purple flowers white, so we can do functional analysis of genes. Here's a homeotic gene, and turning it off turns petals into sepals and stamens into carpels. So we can do functional analysis as well. So again, we've got this incredible diversity, floral diversity and ecological diversity. The species are just really diverse. They're extremely similar, though, across the vast majority of the genome. And this is going to allow us to identify these highly differentiated regions that are apt genes that are important in species differentiation. And the repeated origins of the same traits allow us to study convergence, as I have shown you with flower color. Lots of people to thank, collaborators up here, postdocs and graduate students, and of course DOE has been fantastic, and we really appreciate their support and work that they've done. Thank you.
Thanks, Scott. Question, Sarah? Jeff?
I can't see. Oh, okay. Great.
Scott, hi. So I guess I have two questions.
Okay.
Firstly, with 43 fixed differences out of 3 million SNPs, are they really species? They're kind of not really species, right? They're just giant morphotypes.
Well, I think that's a really good question. When I'm in the field, being a field biologist, these act like a species. I mean, they are ecologically different. They interact with different taxa. They don't interbreed except in these really narrow hybrid zones. So they sure look like species to me.
And then the second question is, I guess your chain of events evolutionarily would be MIB missense mutation, loses color, can't be seen by hummingbirds anymore.
Yep.
And then how long is it until the hawk moth can pollinate it, and how it almost has to have a changed pollination floral structure simultaneous with losing the color? How do you?
Yeah, and there's a couple of things that are going on. I mean, I focused in on the color and the pollinators, but obviously the habitats are really different as well. And so whether there's this correlation with moving to a new habitat and possibly having a more abundant pollinator in that area, which it could be. So I actually think my assumption, although I don't have any data for it, would be is that they've moved into a habitat first and then picked up the pollinator secondarily, is my working hypothesis.
It's kind of like name that tune. Right?
Yeah.
Can you create that species in 43 fixed changes?
Exactly. Yeah.
Any other questions for Scott? Thank you.
Okay, thanks.